welcome in the name of IRISCC to this first webinar on the series related to the research infrastructures included in IRISCC. As you know, IRISCC includes 14 different research infrastructures that are European leading research infrastructures. And today we're going to be introduced to two of them. We're going to start with EIRENE. It's going to be introduced by Jana Klanova from Masaryk University. And then we're going to move to ICOS, which is going to be introduced by Elena Saltykov from ICOS, with headquarters here in Finland. We've been asking to each of the research infrastructures that are going to be part of this series 3 questions. If they could explain to the audience taking part in these webinars, what is the research infrastructure they are representing about, and how does this infrastructure fit in the context of IRISCC? And if they could give us some climate change risks-related cases that have to do with the use maybe of data sets related with their infrastructures. As I said previously, this is a recorded session. We're gonna record just the webinar part about the introduction of the 2 research infrastructures for dissemination purposes, but then we are gonna stop the recording and we are going to move to the questions that the audience might have. And this is going to be the first session out of 7 sessions so that we can cover the 14 research infrastructures. I will put a link to the different registrations for the different research infrastructures on the chat so that you can sign in if you're interested in knowing about the other ones. But without further ado, I'm going to stop sharing and I'm going to give the floor to Jana Klanova. Jana, whenever you want, the floor is yours.
Thank you very much. So now I'll try to share my screen. Going to the full mode. I hope you see it now.
We see it, but we see it as presentation mode. So we can see your next slides and notes.
This should be now.
Yes, now we can see it perfectly.
Thank you very much. So good afternoon, everybody. Thank you for this opportunity to introduce IRISCC infrastructure. You might know that this is one of the fresh infrastructures that was only introduced to the S3 roadmap in 2021. So it means that we just completed our preparatory phase and are getting ready for the implementation phase. And because we say this is the European infrastructure supporting exposome research, let me start with the exposome definition so we are on the same page. We define exposome as the totality of the contacts between the environment and the biological entity. And if we speak about environment, we are speaking about both the natural environment, building environment, and social environment. And of course, climate change, climate conditions, it is part of the environment. So we are very much interested also on how the changing climate is affecting the human health and population health. Another definition that we are using is functional exposomic, which means the systematic studies of the relationships between the environmental exposures and phenotype and health. And I would like to attract your attention to the picture that is in the upper left corner, showing how we relate the exposome research into the genomic research, because phenotype is always given by the genotype and then all this totality of the exposures from the natural and social environment. And as we think about the functional genomics, how the genome translates into the transcriptome, proteome, metabolome, and so on, in all those levels, we can see the impact of the environmental exposures. So they can affect the genome, but also the proteome and metabolome and microbiome in your gut and so on. And in order to study this, we are taking two different approaches. One is bottom up, the other one top down, or sometimes we call it as external and internal component of the exposome. For the IRISCC, it's very important, the external part, because in the external, we are trying to map all the environmental influences, including the climate, and then relate it to the data on human health, either the individual or population. While in the other approach, internal, we are much more focusing on the biomarkers that can be measured in the body, trying to see the exposure markers and link them to the effect markers. And this is something that we can bring also in more comprehensive, holistic research that is now kind of hidden under the topic of the One Health, because now this is one of the big things of the European Commission, that they want to see the holistic approach of One Health. I would say that functional exposomics is really introducing a lot of tools that are needed to have this holistic approach. This is just showing, how the whole field was developed, starting with the first introduction of the topic or the name of human exposome, and then gradually developing the concept. It is not only in Europe. It is very, the exposome community is very active in the US, and we closely collaborate with them. There was a whole bunch of the European and US projects developing the exposome concept in the last 10 years, and it kind of resulted in the establishment of IRIN as the research infrastructure. As I said, it was introduced to the roadmap in 21. Here you can see the membership. We have 25 countries, and our ambition is not European, but global, because at the same time, we are building the global exposome community and the forum, so we really want to link it. For us, it is extremely important that our efforts are aligned, that we have the same quality standards for the measurements, quality standards for the data storage and the assessment. So we are also working closely with the US, Canada, Australia, Japan, and other countries outside Europe. Just to explain what are the main pillars of our services, what we are bringing into the landscape of environmental infrastructures in Europe is the chemical exposures, because this is something that goes across the earth and water and life ecosystems, and none of the existing infrastructures was covering the toxic exposures. So this is the new unique element. So we provide the laboratory capacities to analyze chemical exposures for any metrics, abiotic and biotic. And of course, what we are adding are biological responses. So all kinds of biomarkers of exposure or vulnerability or the effect of these exposures. And this must be complemented with the assessment of hazard, which means toxicology. What are the effects of such exposures? And then the overall risk assessment. And again, it could be environmental risk assessment or human health risk assessment. And then, of course, the huge data part. We provide environmental data, and we work closely with all the other ENVRI infrastructures, hoping that we will be able to connect as good as we can with other sources of environmental data, including climate data, to have this holistic view of environmental exposures. But then we are also linking to the infrastructures from human health and life sciences, because we are dealing with human data. Within our infrastructure, we have many national and international cohorts and cohort projects. So we are dealing with sensitive data, including the genomic data. And then, of course, we are also investing into the data processing tools and building the various types of the models for the interpretation of the data. This is just the illustration showing how complex this field is, because IRIN is in the middle, where you can see those two yellow spots talking about the exposure assessment and then understanding the impact of those exposures. This is where we see this internal and external exposome approach, assessment of hazards and risks. But in order to understand that, we need to have an access to the health records, to the genomic data, to the information about the psychosocial influences. So we are very open to collaboration, not only to the genomic projects, but to the National Institutes of Health Information and Health Records. We are working also with some infrastructures like SHARE from the social and humanity areas to really address also social things. And one of the important tools for us are population studies. And it could be healthy populations or health surveys or clinical studies. And then our impact goes into, in one hand, into the improved chemical management and policies protecting human health from chemical hazards, but also contribution to biomedical innovations and problem-solving capacities. So to link the internal and external factors of the exposome, we need to look into the ecosystems and physical and chemical environment, but also into the behavioral, which is another link to the social sciences, because your life choices, your sleep behavior, diet, exercise, all of those decisions are also important for the final exposome. So now going to the IRISCC project, I said that IRIN is not yet ERIC, so it means we have to step into the InfraServe project as the single entities. Into the IRISCC project, we entered as the coordination entity. So Masaryk University and RECETOX Center is the partner of the IRISCC project. I said that IRIN is very new infrastructure, but this is not true for RECETOX, because RECETOX RI is the large national infrastructure that was placed into the Czech National Roadmap in 2011 already. So we have 15 years of the experience with providing open access services. We've been developing our services gradually in those 15 years, and we are fully equipped. So what we are offering is an access to our accredited laboratories, to our population studies, several generations, to our environmental monitoring networks, but also we are building the information platforms for open sharing of data on chemical contamination. And the most important part that we are offering in IRISCC is the capacities of accredited analytical laboratories. This is why the person you mostly see in the IRISCC meetings is Petra Přibylová, who is the head of our accredited laboratories. So here are examples of the chemicals that we are capable of measuring, and they include legacy chemicals like dioxins, furans, bisphenols, organochlorine pesticides, but also emerging chemicals like brominated flame retardants, various types of novel pesticides, but also perfluorinated compounds, pharmaceuticals, bisphenol, the plastic additives. But as I said, we can also measure metabolites, both metabolites of those exogenous chemicals, but also metabolites that are reflecting the chemical exposures, like the levels of thyroid hormones and so on. So these are the capacities of our laboratories. In addition to that, we are also running the big atmospheric monitoring networks, because this is one of our expertise, is the development of passive sampling devices, both for the air and water. And in collaboration with our international partners, we are since 15 years running the air monitoring networks all over Europe, but also supporting Africa, South America, and some parts of the Asia. In that, we closely collaborate with the United Nations Environment Programme, and we are contributing to their global monitoring plan. And again, this is something we are not specifically offering as a service of IRISCC, but this is something that we can also offer an access to all the sites where we are monitoring. And again, the population studies, we do have the grown-up birth cohorts that were established already in early 90s. We have the new birth cohorts. Those are the children that are just reaching the school age. We also have the aging cohorts, the national seroprevalence study. Altogether, we have about more than 30,000 samples in our biobank that are available for future use, of course, including all the complementary data. So those are the samples that can be used for the assessment of long-term impact or the assessment of long-term trends in the exposure of the human population. That is very important to be linked with the exposure data from the environment. But of course, we are also developing the new type of the markers that would allow us to study the health effects on the phenotype. In the last couple of years, we are also developing the laboratories that are, again, not offered via the project, the IRISCC project, because we also have the biomarker laboratories that can measure quantitative and qualitative assays of proteins and multiplex metabolites. And I said our laboratories are accredited and we are very much focused on the FAIR principles. So all our methods are really reproducible, scalable, and we support open science, but by providing an open access to all the experimental sampling and analytical SOPs, we are openly sharing mass spectral libraries for non-target screening approaches, and we are investing a lot into data processing pipelines, and this is all publicly available. And another thing, we closely collaborate with the IRISCC Infrastructure and their Galaxy environment, where we are building data processing workflows for the analysis of non-target data, high resolution, high volume data from mass spectroscopy that can be used both for environmental and human health samples. And I already mentioned that we are also developing the information systems. This is for the chemical exposures. Again, was something that was missing. So we established the Genesis system that is showing different environmental matrices and their chemical contamination. And this is something that can be, again, in the future linked to other GIS level or other geocoded data about other environmental factors. And this is where I see the opportunity again for the future with the infrastructures like ICOS, Actris, Alter, and others, how to translate your data into the GIS maps that can be layered on the top of each other, and how this can be used for the assessment of the total exposures of the populations living in certain locations. In order to do that, we are also investing into building the urban exposome tools. It means, libraries of different GIS levels, where we can bring together not only data about the air quality, but also about the traffic or green urban places or roads or pollen. And this is something that can be used to interpret the data from the national health registers and look for the factors that might be behind the development of the respiratory diseases or certain health conditions. So again, this is a good opportunity. I already said that we are closely working with the United Nations Environment Program and also with WHO. We are the official WHO and UNEP collaborating center. And we work as partners with supporting the implementation of the UNEP conventions like Stockholm or Minamata Convention protecting health from chemical exposures of organic compounds or mercury. So we are also active at the conferences of parties. We are organizing a lot of joint meetings and training programs for the parties. And our information systems are the official reporting systems to collect the global data for the global monitoring plan. And also we very closely work with the group of Earth observation contributing or strengthening the European contribution to GEO and Copernicus. So what I was talking about, it was a bit broader than ICOS, and even though, these health focused research area don't seem directly related to this project. They, in fact, are very much related because you are also interested in an impact of all the climate change and climate conditions on the health. So what we are bringing here is really the tools that can help linking the environmental with the health data, either on the population level with the health registers or on the individual level. Thanks. That should be all from me. Here are a few pictures from the Brno city where the IRIN headquarters are seated. You probably recognize the picture of Gregor Mendel because this is the place of Gregor Mendel, and I hope you will have chance to visit this city in the future. Thank you very much.
Thank you very much, Anna, for your comprehensive presentation about IRIN as a research infrastructure. I think we can move forward to the next research infrastructure. If someone actually has a question about IRIN, they can also write it down in the chat, and then we can catch it up. Or you can wait till the end and we can have the session of questions. But without further ado, I'm going to just give the floor now to Elena Saltykov from ICOS. Elena, the floor is yours, and we can see your presentation in presentation mode.
I'll unmute myself. Yes, good afternoon, and thank you for this opportunity to tell you a bit about ICOS and what we do in IRISCC project. And I've split my presentation in 3 parts. I start by telling you what ICOS is. Secondly, I'm showing you super briefly something about what we do in IRISCC project and more about general philosophical approach, why we are so very interested in IRISCC. And then I have 3 practical examples from past, how ICOS can be used in relation to the climate risks. And ICOS is short for Integration Carbon Observation System, and it provides standardized, high-precision, open data from 180 measurement stations across 16 European countries. And at those stations, we are measuring greenhouse gas concentrations. We are measuring carbon fluxes in 3 domains, so fluxes between the atmosphere and ecosystem. This is both the ecosystems, the plants taking carbon from the atmosphere, but also the ecosystem respiration, breathing out CO2. As you know, all of us are breathing out CO2, and we measure that as well. And the same way we're measuring the fluxes between the atmosphere and ocean. Actually, the parameter we measure the partial pressure of the CO2 in the near surface of ocean water. That's going about as technical as I'm going to go with that. What we produce is that we produce long-term data sets, which are covering then the entire carbon cycle. You can see the carbon cycle in the picture on the right. I have more pictures of that. So we're talking about how the carbon is moving between the soil, the ecosystems, the atmosphere, and the oceans. And I made in bold the words long-term data sets, because when you study climate, you want to have data sets which are much longer than one project or one PhD thesis. You want to have years and years of accumulating data sets. And this is coming back to the first bold word, standardized. Because we have data which has been collected by people who are already retired. And we can't phone to them and ask that, what do you mean by T1 and T2 in your Excel file? But everything is going according to the protocols which we have agreed between the station PIs so that they can be implemented in all of these 16 countries. And we make sure that whenever new stations or principal investigators are joining the network, they will follow the standard. We don't accept their data if it's not according to the standard, and this is where we are strong. And together we're building an ICOS environment. It's not only us 20 people in the head office, it's roughly 800 scientists, which are involved in over 200 institutes throughout these 16 countries. So why do we matter? I'm saying we're very important, all right? The work we do and the data we collect is helping humankind to quantify sources and sinks of greenhouse gases. We'll know the picture in the previous slide where there's arrows going up and down, but then how much, how many kilos? This is where we are becoming essential. And although the data is improving our understanding of the carbon cycle and the Earth system in general. And when we offer a harmonized data set across Europe, then we support scientific research, climate change modeling, and evidence-based policy and decision-making. And these long-term consistent observations, they enable detection of trends, changes in fluxes, and extreme events, which is a key for assessing climate change impacts and mitigation efforts. It's a common thing to say when you talk about people losing weight that if there is no scale, then there's no diet. We want to be that scale to be sure that the mitigation continue when you see that it has an effect. And as I said, the data we have is open access. For researchers, we do have the ICOS Carbon Portal where all our data is open. And even though we're measuring in very different places, different environments, it's one place, it's one address where you go. And then you select, this is what I want, these are the years, these are the stations. And it comes your huge file of data. If you don't want to do that, we also have Jupyter notebooks which allow you to do some calculations on our servers. We provide you the computing space. And when you have made your testing, then you might know, yes, this is what I want. And you can continue with that. And we allow researchers, but we would also like to have policymakers, educators, and other users to download and use the data. But of course, it's the license we have is CC BY, which means that when you are using our data, you are required by the license to also acknowledge and cite those who have provided the data. We also have now something which is relatively new, a quick visualization of atmospheric CO2 trends. And we have a tool for that, which is called the ICOS curve. And it's showing daily, it's updating every day data which was measured yesterday. So you can really see this horrifying curve, which you might know from Mauna Loa as a Keeling curve. But we do this for Europe. In the picture on the right-hand side, I have there in blue the line from Puijo, a station in eastern Finland. But then I have there the gray shades, which is showing all of our atmosphere measurement stations. And you can see here the all too familiar trend that each year there's more and more CO2 in the atmosphere. And that is the matter which is causing climate change. It only the amount of those greenhouse gases in the atmosphere are the things which are affecting the temperature, not the promises what the governments do, not the plans of mitigation, but the amount of CO2 and other greenhouse gases are the ones which are linking to the climate change. You can also see that even though each year there's uphill and downhill, the downhill is when the plants are taking carbon from the atmosphere. The uphill is when they are not taking any atmosphere, but the leaves are rotting, and also that's usually when there's a peak in the human emissions. You can also see that years are similar, because the ecosystems and people behave differently, different years. And you can see that they're spread because different parts of Europe have different amounts of CO2. And all this we're measuring every day, every half an hour. We are doing this measurement and we're sharing the data with you or with anyone who is interested. What we do in ICOS practically, we're working on soil carbon service design lab in which we are exploring new services. And the focus here, unlike in majority of the work we've done before, is focusing on non-academicians and policymakers to tackle the climate risks. The work we do here in IRISCC is related to carbon removal certification framework, which is currently in the works of EU Parliament, and we're working with the Parliament's group for that topic. As a first step, what we're doing is that we're bringing a CROP21 dataset to ICOS environment to better support the comparisons of the field data at agricultural sites, and then we're starting collaboration with the team which created this dataset for 21, and we hope to develop a new dataset called CROP26. If you want to know more about this dataset, which is related to agriculture, I have put here a link to a poster by that team. But I'd like to say a bit broader picture that why this partnership matters. This is one of the projects where we're really putting effort in ICOS. And what we bring to the family is that we provide the carbon cycle foundation for IRISCC. We're delivering the harmonized high precision greenhouse gas and flux observations, which are essential for climate risk assessment. But then we see that IRISCC is integrating ICOS data with other environmental and socioeconomic infrastructures, which then enable multi-hazard and cross-disciplinary climate risk analysis. And then together, ICOS and IRISCC bridge science and decision making. We offer a robust observational evidence, and then IRISCC project, and we as part of that project as well, are transforming it into accessible services, tools, and knowledge for adaptation and mitigation. And we see that this partnership enhances Europe's climate resilience capacity and is supporting informed policy, scenario analysis, early warning systems, and long-term sustainability planning. Match made in heaven. I'm now going to show you 3 case studies from different sides of the carbon cycle. I've shown you half of a case study already because I showed you how the concentrations in the atmosphere have been changing in Europe last 10 years. That's the ICOS curve. And I do again encourage you to go and find that curve. It's somehow addictive to click different options at different stations there. But we do more than that. And I like to say that this kind of monitoring infrastructures like ICOS, they are essential foundations for understanding the real world carbon cycle. The carbon cycle dynamics, under stress. So when something is happening, and the stress can be caused by human or it can be changed by climate. And the climate action and mitigation plans, adaptation plans must consider this variability. It's not enough to track emissions. We must also track how things are responding to the extreme. In the picture, which is from Wikipedia Commons, we have there on the right-hand side, we have their emissions. Some of them are coming from the factories and cars and fossil fuels, but also cement production and other anthropogenic sources. There's also, of course, natural sources. As I said, breathing is very natural. Then there are the sinks on the left-hand side, and then the ocean part. And I have one example where the emissions were decreasing, and 2 examples linked to each other where the sinks were decreasing. first case study is related also to the first talk of today, health. COVID happened, not so past history. And I have here a study by Giacomo Niccolini and his colleagues related to the reductions of the CO2 during the lockdowns. The gray dots are showing a number of European cities and how much were their we can just say that these were emissions. So relative flux change is the zero line, that's the average of all the cities. The gray was before COVID, so-called normal times. Red dots happened during the lockdowns. The more you on the right-hand side, the more strong limitations that city had about people meeting and driving to the office. And then the biggest danger was over. And then the limitations were let a bit looser. And yes, we can see that the emissions increased, but not to the pre-COVID levels. And then second moment, even more looser limitations about what people can do, still not coming to the pre-COVID level. The humankind changes habits and looks like it changed permanently. I can just think about this webinar that most likely before the COVID, we would have all been traveling to one city to look at these PowerPoints. While today I haven't left my home except by walking. I haven't had much less emissions that I would have had if I had traveled anywhere. And in this study, the data revealed dramatic reductions of urban emissions between 5% and 87%, depending on city and area. And in several cities, the emissions stayed surprisingly low, even after the restrictions were eased, underscoring the sensitivity of urban carbon fluxes to human behavior. And the implication is that these observations offer a natural experiment insights into what emission reductions from lifestyle or policy shifts might look like. But to me, this is also underlining the thing that we didn't know that COVID is going to happen. And yet we were, luckily enough, we were measuring already so that we have the gray dots, something to compare that how were these places before those limitations come. It's super easy to get funding to a project when something dramatic is happening, but you should have also the control data before that catastrophe happened. And if anybody is from EU planning about funding, this is a message for you. If anybody of you is lobbying in Brussels, this is also a message for you. Second case I have is maybe the most famous ICOS study. In 2018, in the end of summer 2018, we had a science conference, and there everybody was trying to say that, hey, in my city, it has been super dry and the ecosystem is dying and we're measuring very little carbon uptake. And then they agreed, this was before I joined ICOS, they agreed to make a joint study group. 200 people from different parts of ICOS community joined. They shared the data, they shared the initial findings, and then it became a special issue in philosophical transactions of the Royal Society. I'm showing you just one picture, which is listing all the then ICOS atmosphere stations. So each line is one station somewhere in Europe. And while I said that it's ICOS stations, there are a few other which were not yet member countries. And we can see that, so the horizontal axis with awfully small numbers are the years starting 2008, 10, 12, 14, 16 and 18. And you can see that here every year has a cycle that in summer when plants are growing, values go blue, there's less carbon in the air, and then in winter they're going red. And then comes the summer when the carbon uptake has been much smaller. And that was because it was so wildly dry. If you want to zoom to one or two stations, I have another publication from the same journal. I also need to show this as this was done by the IRIS coordinator when he was a young man. And so I have there two stations, and on the left I have the daily CO4 fluxes up or down. In gray, it's the reference period, so before this big drought happened, and in black is the year 2018. And it's not that dramatic when you look at the daily values, but when you look at the cumulative net, you can see that the area between the gray line and the black line, that's all about the carbon which did not go to the ecosystem, which was not taken up by the wetlands of Myklemossen and Sikaneva, because it was so dry that the sphagnum was not growing there. And this is another example of how we can show that this is not a local effect, this is everywhere. And we can even quantify that at this specific place, this much carbon was staying in the atmosphere. This is why the graph in the previous picture did not go blue, because the carbon did not go to the ecosystem. I have a more quantifying study from a few years later in 2022. Again, it was dry. More model data was used here so that they could do more quantifying analysis. The browns in the picture show the areas where the land was, soil was extremely dry. The darker the brown, the darker the brown, the drier the land. And you can see that it was severe and it was widespread. And these people, Van der Woude and his colleagues, they published a Nature paper, and I have here the link as well. They could quantify it, that the carbon uptake, which didn't happen, was between 56 and 62 teragrams, or 56 to 62 billion tons of carbon. This number might not tell you that much if you're not a specialist, so I want to compare it to the emissions of entire Finland in 2024, excluding the LULUCF. That was 39 teragrams. So 1.5 times emissions of Finland were just not going away because the trees died. And these studies, I've shown you 3 studies, and they show that climate-induced extreme events like droughts and heat can undermine the role of land ecosystems as carbon sinks. And that fragility is highly relevant for climate projections and mitigation policy. Because, of course, when there is more climate change, our ecosystems are not adjusted to that. It's more likely that trees will die either for flooding or for droughts or for forest fires or all those climate risks we're studying in the IRISCC project. So why these results matter for the climate risk assessment? So firstly, these results reveal how quickly and strongly greenhouse gas fluxes respond to changes either in human behavior, as I showed you in the lockdown picture, or in environmental stress, as I showed in the 2 drought studies. Many of the impacts of climate change are so slow that they don't come to headlines that fast. That's why this kind of small catastrophes are easier to get through the threshold of media. These also show that the carbon sinks, like forests and other ecosystems, are vulnerable to climate extremes, which might reduce their buffering capacity just when the emissions remain high, or even if they grow. And these results also provide empirical evidence. They're not just model projections, which some denialists have some challenges to trust, but these are empirical evidence. There are some measurements, which is then strengthening the credibility and the scientific basis for climate risk assessments, mitigation planning, and ecosystem resilience policies. And this was my last slide, and I'm ready to take your questions. Thank you.
Thank you very much, Elena. That was a very, very nice presentation. I want to thank both Jana and Elena for sharing their time today and showing to us their research infrastructures in this webinar series, the first one out of 7. As we said, it was very nice also to see how they relate to the IRISCC and the services in relation to climate change risks. As we said, we are going to stop now the recording, and we're going to take some questions from the audience.